All right, let's dive into a really powerful scientific technique. I want you to think of it kind of like molecular Velcro. It's used to sort the essential ingredients for pretty much everything, from brand new medicines to making sure we have clean water. It's called ion exchange chromatography.
So to really wrap our heads around why this is such a game changer, we first have to understand a huge problem that scientists are up against, well, every single day.
Just imagine this. You're trying to create a new drug, and you need to pull one single specific protein out of a super complex soup, like blood. Or maybe you're trying to find a tiny, dangerous contaminant in a food sample. It's the classic molecular needle in a haystack problem, right? And solving it is absolutely crucial for pretty much all of modern science. So how in the world do you find that needle? Well, scientists came up with this incredibly elegant solution that basically uses a molecule's own electrical charge as a tiny handle to just grab onto it.
And that technique is called ion exchange chromatography, or IEC for short. The idea behind it is actually surprisingly simple. It all comes down to basic electrostatic interaction. Think of it like a very, very controlled form of molecular static cling. Things with opposite charges stick together, and things with the same charge push each other away. That's it.
So the sorting machine itself is a column, and it's packed with a solid material that we call the stationary phase. Now, this is usually a resin made of millions of tiny little beads. And here's the key. The surface of these beads is coated with charged groups, creating this sort of electrical gauntlet that the whole mixture has to run through.
And that brings us to the two main flavors of IEC. First, you've got cation exchange. In this case, the resin beads are negatively charged. So what do they grab? The positively charged molecules, the cations. Then you have anion exchange, which is just the opposite. The resin is positively charged, so it snags the negatively charged molecules, the anions. In either setup, any molecules with the same charge as the resin just get repelled and wash right on through. See? It's all about opposites attract.
Okay, so we've got the principle down, but what does this actually look like in a lab? Let's walk through the machine itself and see how this whole separation process happens.
So a typical system has four main parts. You've got a pump that pushes a liquid through the whole setup. Then there's an injector, which is where we introduce our sample mixture into that flowing liquid. From there, the mixture flows into the column. That's the heart of the whole operation, where the actual sorting happens. And finally, at the very end, there's a detector that senses the molecules as they come out, one by one. And here's how that all comes together in 5 steps. First up, equilibration. We're just prepping the column with a special liquid. Next, sample loading. We inject our mixture. Then comes the magic part, binding. The molecules we want, the ones with the opposite charge, stick to the resin. After that, we do a wash to get rid of all the impurities that didn't stick. And finally, the whole point of the exercise, elution. We change the liquid to release our target molecules from the column so we can collect them.
But get this, it's not just some simple on or off filter. The real power of this technique is how you can fine-tune it. Scientists can take precise control of the process to separate molecules that have even the tiniest differences in charge. So, how do they pull that off?
Well, they basically have 2 main control knobs. The first one is pH. By changing the pH of the liquid flowing through the column, they can actually alter the electrical charge of a molecule, like a protein. That lets them control exactly how strongly it's going to stick to the resin. The second knob is salt concentration. By adding salt, you introduce a flood of tiny salt ions that start competing for those charged spots on the resin, and they effectively knock our target molecules right off so we can collect them.
And this is where the real purification magic happens. During that last elution step, scientists don't just dump in a bunch of salt. No, they create what's called a salt gradient, which means they slowly and very precisely increase the saltiness over time. Molecules that are only weakly stuck to the resin get kicked off first when the salt level is low. But as that salt concentration climbs higher and higher, the more strongly bound molecules finally let go. It allows for this incredibly precise step-by-step separation. Okay, so this is all really cool science, but why does it matter? I mean, why is this ability to sort molecules by charge so important? Well, it turns out it has a massive impact on our daily lives. The applications are literally everywhere. That water softener in your house? It's using ISC to pull out the calcium and magnesium ions that make water hard. It's a guardian in the food industry, detecting dangerous stuff like arsenic or heavy metals before they ever reach our plates. It's the absolute workhorse of the pharmaceutical industry, purifying the proteins and antibiotics that go into life-saving drugs. And in the hospital, it can help diagnose diseases just by analyzing a blood sample. It's amazing.
And that really sums it all up, doesn't it? From huge protein molecules all the way down to the tiniest amino acids, ion exchange chromatography is just one of the most powerful and versatile tools we have for separating charged particles. It's a foundational technique that makes so many other discoveries possible.
It really makes you wonder, doesn't it? With a tool this precise, a tool that lets us sort the very building blocks of life and matter, what new cures, what new materials, or what new solutions are just waiting to be discovered? What's the next thing we'll be able to separate?